So today we'll be talking about structural mass spectrometry. And I'm going to start by defining what that is. And we'll have a look, first of all, at what you can learn by structural mass spectrometry. And we will say something about ion mobility. I'm not going to be covering native mass spectrometry. That is a structural biology technique. We covered that in some detail last week, so we won't go over that again. I'm also going to talk about limited proteolysis, some structural information you can get from chemical derivitisation, hydrogen-deuterium exchange, which is a very powerful technique, and also cross-linking mass spec. So to begin with, I've taken this definition, I think, from Wikipedia. Structural biology is concerned with the molecular structure of biological macromolecules, how they acquire the structures they have, and how alterations in their structure can affect their function. And if we picture structural biology as this flower with several different petals or branches to it, structure determination is one branch of structural biology. But there are others, and I've listed them here. There are various pieces of structural information that are all going to be of interest to the structural biologist. And all of these different pieces of information are tractable by mass spectrometry. And please do not think of mass spectrometry as a single technique. It is a range of different techniques, and they share the same instrumentation, but the approaches to these techniques are all different. And how you should be viewing mass spectrometry is this multifunctional tool. And you can use this tool in lots of different ways to get the structural information that you're interested in. So I'm going to make a start now with ion mobility. So ion mobility is used in mass spectrometry to determine shape and also size of gas phase ions. So how ion mobility works, and we have mentioned this before, so we need a drift cell within our mass spectrometer. Here it is, and this is filled with a low pressure gas, and our ions will drift through this cell under the influence of an electrical field. So what will happen is at low velocity they will collide with the gas molecules in this cell, and they will be retarded. And the more gas molecules they collide with, the slower they will be, and the longer it will take them to traverse this field. The number of gas molecules they collide with depends on what's called their collisional cross-sectional area, or the CCA. So the larger the collisional cross-sectional area, the longer the drift time will be. And ion mobility has been around for a very long time. It has been incorporated into mass spectrometry, I would say, for about the last 20 years. So it's built into the ion optic path. And in theory, having an ion mobility cell will give you an additional data dimension and an additional separation dimension. I say in theory. There are some issues with ion mobility. The resolving power of ion mobility, I will make an exception with Timstorff, but most ion mobility cells, the resolving power is quite low. You may be able to resolve 10 to 15 peaks, not a high resolving capacity in comparison to other separation techniques. You do need to calibrate an ion mobility cell if you want to derive collisional cross-sectional area. That in the past has been problematic. I believe it still is. So what happens with ion mobility data is you get something like this. So you have an MZ, the MZ values are plotted against the ion mobility drift time. And the intensity of the ions are represented with different colors. And so this is typical piece of ion mobility data. This is called a drift scope plot. And I have to say, it does look incredibly impressive. It doesn't look anything like a mass spectrum. It looks totally different, and it looks like you're getting lots of information here. Well, there is lots of information on there, but that information doesn't really add very much to what you would already get from the spectrum. What I would say is the first ion mobility QTOF was launched by Waters in the about 20 years ago, and they use this very successfully as a marketing tool because it does look like you're getting new information. However, there isn't really anything new there, and I just want to explain that. This is a piece of data from a paper some years ago, and what they've done here is this is your ion mobility data, this is your drift time versus mass to charge, and on top of that the authors have overlaid the spectrum. And I stared at this for a very long time, and I realized something really quite important about ion mobility data compared with mass spec data. What I did with this is they've given you the mass to charge. We know the mass of the complex, so you can assign charge states onto these peaks. And what you can also do is you can plot the charge state against the drift time. And I've done that. And if you do that, what you discover is the drift time and the charge state, there is an inverse correlation. And that correlation is exceptionally close. What that means is if you take ion mobility data and you correct it for charge state, it's the same as the spectrum. These two pieces of data you're looking at are the same. There is no difference. So you can use ion mobility, and it is used. It gives you useful information that you can't get any other way about unfolded and denatured proteins. So you can get collision collisional cross-sectional area. You can derive that from peptides and from denatured proteins. But in general, of course, this is not what structural biologists are interested in. Most structural biologists are interested in natively folded proteins. And if that's what you look at, what you discover is the drift time and the mass to charge always correlates, and there is always a straight line. Here's an example of one. Here's an example of some more. Here's some more. Every time I look at native data in ion mobility, you always see this straight line relationship. This is a piece of data from Agilent. They make ion mobility mass spectrometers, and this is their data, and this is the most perfect correlation I have seen. So this is a myoglobin subunit, and you can see that correlation is not going to get any better than that. So what that means is if the drift time is measured for a native protein, you can calculate the mass to charge and vice versa. So what this actually means is for native species, the drift time measurement doesn't actually generate any additional information, and it's pointless. If you work in ion mobility, I apologize for that. If your company makes ion mobility mass spectrometers, I apologize for that as well, but that is what you see. So I would not recommend ion mobility as a tool to study structure, particularly. I know a lot of labs do that. My preference would be for native mass spectrometry. It is a structural tool. We've covered that in the previous session. Something more interesting, I think, is the use of limited proteolysis. So as I'm sure you're all aware, proteins have domains. So there are stable structures within proteins and then usually domain linkers. And these linker regions are very often more accessible to protease digestion than the more stable structures like an alpha helix. So we can use mass spectrometry, intact mass analysis and MSMS analysis to study domain boundaries. And how we do that is very simple. The protein is subjected to limited proteolysis, either using trypsin or chymotrypsin, at a very low molar ratio. So you sample it over time. So normally, in MSMS experiment, we are looking for complete digestion. In a limited proteolysis experiment, we're aiming for the opposite. So what we want to do is we want to catch the digestion midway. And if you sample at different times, what you can see is there is a stepped function in the digestion. It doesn't go smoothly. Part of the protein is highly susceptible to protease action, and then the domains, the stable domains, are more protease resistant. So they will generate this resistant fragment in that you can see on the SDS page. So what we then do with this is you would take this fraction and run it on LCMS. You'd obtain an accurate mass spectrum, and ideally you would get a fragment corresponding to this peak. And then with some very simple software, which I've already shown you, you can match that fragment to an internal sequence within the protein sequence. And you should be able to determine the domain boundary, so where that the two boundaries of that domain. What you can also do is you can excise these bands from the gel and subject them to tryptic digest LC-MS analysis, and you will get partial sequence coverage for these, and then you can try and correlate that sequence coverage to the fragments that you've identified by intact mass. So very often we need to use both of these techniques to locate our domain boundary. And that can be very useful in structural biology, particularly if you want to develop, make constructs, stable constructs for crystallization. This is a very good way of working out where those boundaries should be. Another thing we can do with mass spec is we can measure intramolecular disulphide bonds. So we're not going to get this automatically from the MSMS data. Here's an example of a protein we did this for. This is a membrane protein, PKD2A. We got the crystal structure for it, but the crystal structure does not tell you everything. You can see where the cysteines are, and you can see that there are two cysteines in very close proximity to each other. But what you cannot tell from the diffraction information is you cannot tell whether that is actually a disulfide bond or whether these cysteines are just in close proximity to each other. So you can use MSMS to determine that. And how we do this is a bit complicated. But if you take an example here where we have the blue peptides, peptide A and B, peptide 1A, 1B, they are joined by a disulfide bond. If you take the example of peptide 2A and 2B, they are in close proximity, but they are not, that bond is reduced. So what we need to do is we need to duplicate that. So we have these two separate samples, and one sample we subject to reduction and alkylation, and the other sample here is untreated. If we then add iodoacetamide, so that is going to alkylate any free sulfhydryl groups, we digest with trypsin and we subject to LC-MSMS, and then we're going to look for peptides. And the possible peptides we could get are this cross-linked peptide here. 2A and 2B are unlinked, or we could have 1A and 1B unlinked, and 2A and 2B also unlinked, but they have been alkylated with the alkylating agent. So if you do this analysis and you look for these peptides, if this peptide, this peptide disappears in the untreated group, but it's present in the reduced and alkylated group, that tells you that it must be disulphide bonded. So detection of peptides A and B following reduction and alkylation, and the absence without it, that indicates the presence of a disulphide linkage. And so this is the data that we did. We did a number of different digests using different proteases. We weren't able to get full coverage of PKD2A, but we were able to get coverage on the part of interest. And here we were able to determine that this linkage, C161, is actually unlinked. So the MSMS from non-reduced tryptic digest shows that cysteine 161 is unlinked. Now that analysis took quite a long time. We had to use different enzymes and several MSMS analyses. There is an easier way of doing that. And the easier way of doing it relates to some data I showed you earlier. So when we were looking at native mass spectrometry, I showed you this plot where we've taken the maximum charge state, so that's this value here, for a range of different proteins, and plotted it against the residue number. And you can see we have this straight line. If we reduce these values, they will fall on that line. What we can do is we can take PKD2A and we can plot it on this graph. And if we do that, if it falls on this line, we can conclude that all the cysteines in PKD2A must be unpaired. And that analysis time is about 10 minutes. So another thing we can look at, which gives us lots of information on the structure and function of a protein, is solvent accessibility. And the way we do that is by hydrogen-deuterium exchange. So the principle of the technique is we take our protein and we incubate it with deuterated water. And what will happen is there will be exchange happening across the backbone hydrogens of the protein chain. And this exchange will be particularly rapid where that backbone is exposed to water. So the solvent accessible backbone of the protein is going to be heavily deuterated. So this is what happens. Here we can see the exposed regions of the protein are deuterated, shown in red. We then need to quench the reaction. So we put it into an excess of non-deuterated water. And we have to then maintain the pH at 2.5 degrees, and we have to pH at 2.5 and the temperature at 0 degrees. And the reason for this is that you can get back exchange. So the deuterium can be exchanged back for regular hydrogen. So you want to minimize that back exchange. We then submit the protein to digestion. We need to use pepsin. The reason we need to use pepsin is because we have to keep the pH low. So pepsin is active, pH 2.5. That digestion will happen at 0 degrees, so we need rather a large amount of pepsin. And so we're then going to end up with these, I'm just going to get a different pointer, just a minute. Okay, so these, so we're going to have a mixture of peptides. Some of these peptides will be deuterated, and some of them will not be. Some of them will be partially deuterated. We then need to separate those peptides by chromatography. It has to be fast because remember, back exchange is going on all the time, so we want to do rapid chromatography. We need to keep the pH at 2.5. We need to keep the temperature at 0 degrees. And we want to do that in microflow or nanoflow. That's actually quite difficult. HPLC does not really work very well at low temperature. It tends to increase the back pressure. So you need special columns, special chromatography to do this. We will then generate, we need to generate high resolution MSMS data, and the hydrogen-deuterium exchange is measured as a result of a mass shift. So we will end up with a range of peptides, some of which will be deuterated and some of them which will not be. And the deuterated peptides, we can map those onto the sequence, and this has been done here in red. So these are the peptides where exchange for deuterium has been fast, and here, and the blue ones, this is where exchange has been slower. So this is the linear sequence of our protein with these fast exchanging and slow exchanging regions mapped onto it. And then, of course, you can map that onto the three-dimensional structure. And this should be able to tell you where that solvent accessibility resides. So that's the theory. In practice, it turns out to be, it's quite a difficult technique. I think you can appreciate why. You require precision experimental timing. You have to keep these pH optimum at 2.5 and 0 degrees to minimize a back exchange. Pepsin digestion is not too much of a problem. Rapid LC at 0 degrees, as I said, is rather difficult. All this really is achieved with automation. And you can, if you want to, you can do this with a stopwatch and putting your LC column in crushed ice. But it's not really very reliable. It's much better to do this with specialized hydrogen-deuterium exchange automation. This is a robot from Leap Technologies which does that. So you really need to have this dedicated robot, and you also need to have a very sensitive, high-resolution mass spectrometer to extract your data. It's not a simple thing. It does require specialized analysis software. It requires dedicated equipment, specialized equipment, and I would say it requires dedicated personnel. So there are labs that do this, but it tends to be a rather specialized thing. Another way of looking at solvent accessibility is by using ETD. We discussed ETD, I think, in the second or third lecture. So ETD is electron transfer dissociation. This is another method of causing fragmentation that can run alongside a CID. So ETD, it relies on, it's a method for top-down fragmentation. So you can apply ETD to intact proteins and they will fragment, but where they fragment depends on their structure. So what you generally see with ETD data is you will see data from the N terminus of your protein or from your C terminus, but you can also get fragmentation data where your sequence is close to the surface, because the residues there are more labile. So this is an example of using ETD to measure solvent accessible surface area. These are the parameters applied to the collision cell, and where we have sequence data, this is marked in these bars, and here this is the solvent accessible surface area. This is your linear sequence, so from residue 0 to residue 70, and the solvent accessible surface area is marked with these grey bars, and you can see there is a fairly close correlation between the ETD fragmentation data and the solvent accessible surface area. There is another way of obtaining solvent accessible surface area data, and this is again by using derivitisation. So what we've done here is we have taken a protein and we have, this is the unmodified, we just measured the intact mass, this is the unmodified protein, and then we've alkylated it. And the alkylation, this will happen on any unpaired cysteines, and this will result in a mass shift. The mass shift is plus 59, and we can then use LC-MS-MS to subsequently map where these mass shifts are. So this is an example of using chemical derivitisation to determine solvent accessibility. Another very important structural mass spectrometry technique is cross-linking mass spec. So what this enables us to do is where we have protein interaction, it allows us to measure the proximity of side chains between one protein partner and another. So what we need is we need to take our protein complex and we need to expose it to a bifunctional cross-linking reagent. Okay, so this is a cross-linker that can react on both ends. We then need to take our complex and digest it with the protease, and then we do LC-MS-MS. And what we're looking for is we're looking for hybrid peptides. So we're looking for a peptide that has the cross-linker with a peptide from one of the binding partners linked by the cross-linker to a peptide from the second binding partner. And that will reveal a physical proximity between these two side chains. So this is how it works. This is our protein-protein interaction. So we've got two partners, blue and red. We add, so that they're in the ratio of 1 to 1. Then a ratio of 1 to 1, and we have our cross-linking reagent. We can have a light cross-linker. We can also have a deuterated or heavy cross-linker, and they will react with our complex. We then subject the complex to tryptic digestion, and you can see we have a range of different fragments. So some of these have the cross-linker alone, like this one. This is monolinked. You can have a link between two peptides within the same protein. Here's link between two peptides with the other protein. But what you really want to see is you want something like this. So here we have a link, we have our cross-linker, and we have a link between partner A and partner B. But you'll appreciate that the number of cross-linkers in this desirable conformation that we want here, with fragment from both binding partners, is actually quite small in relation to all of the cross-linked fragments. So we can enrich if we for cross-linked peptides. We then need to perform LC-MS-MS, and we can identify which peptides are cross-linked by the fact that they will have this mass shift due to the deuteration, and then we can do our data analysis. And these are the different possibilities we have for cross-linking experiment. What you need to do is you need to optimize your cross-linking. There are different cross-linking reagents. There are quite a lot of different cross-linking reagents you can use. Typical one would be BS3. You can, so here we have an STS page analysis of cross-linking optimization experiment. These are the binding partners, unlinked. So these are molecular weights on a gel, and this is what that gel subsequently looks like following incubation with BS3 at room temperature, at 0 degrees and at 37 degrees. So you can see we have a mass shift, we have these higher molecular weight species, which are your cross-linked proteins. So as I said, the cross-linkers, they have to be bifunctional. They're usually lysine specific. Here's an example of BS3. The length of the spacer arm is quite important because the two lysines that you choose that you are attempting to cross-link, if they are too close together or further apart, this cross-linker obviously is not going to work. So we use, as I said, we use the light and heavy deuterated cross-linkers to identify which peptides are the cross-link peptides. We have to optimize the cross-linker ratio and the protein complex ratio by STS page. We are going to generate large number of species, and so the complexity of a cross-linked digest is actually extremely high. Unfortunately, the efficiency of this cross-linking reaction in terms of peptides from different binding partners that are cross-linked, they may be very low. So it's going to be less than 1%. It may be less than 0.1% of the total number of peptides. There are enrichment techniques for cross-linked peptides. In general, you're going to need a long LC gradient to separate them. You need high sensitivity, so you may want to do this with nano LC-MS. You will definitely need high resolution mass spectrometer. So I would recommend an Orbitrap or a very high resolution TOF. To do your data analysis, you need to generate a database which contains every tryptic peptide from every subunit cross-linked to every other possible tryptic peptide from the other subunits. And just to bear in mind also that our fragmentation data is going to be different. So if we have inter and inter-cross-linked peptides, they have 4 termini rather than 2, and so they will generate more complex spectra. So here's just an example of some cross-linking data where you can see this is you can see we have you can infer cross-links in these different positions, and here's another example of a dimer where you can infer where the contact points of that dimer are. And here's another example of cross-linking. You can see we can infer that these lysines must be in close proximity to each other. Just one more thing I want to say about cross-linking, and this is to do with de novo structure determination. So most structural determination is done by experimentation. So this is typically done by X-ray crystallography or by cryo-electron microscopy. It is possible to determine structure de novo. You can do this on short stretches of amino acid sequence. You can predict it quite reliably over short distances. The problem is, over longer distances, there are just far too many bond angles, and the computational problem is too vast. So currently it is not possible to predict the structure of a protein just from its linear sequence. However, if you have cross-linking data, what cross-linking data does is it introduces constraints into your model. So the more constraints you can introduce, this helps you solve this problem. So if you have a sufficient number of constraints to your model, it is in theory possible to determine the complete structure of your protein de novo. So you don't necessarily need to have crystallography data or cryo-EM data. It is in theory possible to do this. I would say that cross-linking mass spectrometry has some way to go before this is possible, but it is a theoretical possibility. So I just want to summarize what we've talked about today. In structural mass spectrometry, all aspects of structural biology are, in my opinion, amenable to MS analysis. You just need to know what is possible. I would say native mass spec is the most powerful tool available to do this. We discussed that last week. Ion mobility is very widely used. I've not seen anything that I couldn't also do by native mass spec. and I would say it's only of limited value. We talked about limited proteolysis and how we can use that to establish the domain boundaries within the protein. We've looked at a number of different ways of using chemical derivitisation. So we can use this to derive the oxidation state and the linkage, and also solvent accessibility, things like cysteine residues. We talked about hydrogen-deuterium exchange. It's a very important technique. It's very powerful. It's not particularly accessible, but there are labs that will specialize in this. Cross-linking mass spectrometry is an emerging technology. There are new methods coming out all the time. There are new cross-linking reagents becoming available, and it is a very powerful tool when you have proteins and protein complexes that you cannot get the structural information any other way. And in theory, cross-linking mass spec allows de novo structure determination. That's all I want to say. Next week, we'll be looking at how we can use mass spectrometry in the analysis of integral membrane proteins, and I'll be very happy to answer any questions.